Good afternoon, everyone. My name is Phaedra Henley, and I'm a PhD student in the Department of Pathology. Today I'm going to be talking to you about some of the results from one of our large ecosystem health projects set at Lake Naivasha in Kenya. The title of my talk is Real versus Perceived Risk of Pesticide Exposure in Naivasha, Kenya. So Lake Naivasha is the largest freshwater lake in Kenya. It is home to a massive floriculture industry that covers 4,000 hectares, employs up to 100,000 people, and supplies 35% of the cut flowers in Europe, making it one of Kenya's top foreign exchange earners. Migration into this region for employment has also caused a massive population boom. It is for these reasons there are many ecosystem health concerns at Lake Naivasha, including a collapse of the fisheries industry, the creation of large unplanned settlements surrounding the lake, as well as potential exposure to pesticides. Dire ecosystem health concerns has both local and international advocates and research groups pointing fingers at the booming floriculture industry. Our ecosystem health team has adopted a comprehensive, innovative, and transdisciplinary approach in order to assess the socio-ecological system and the effects of the health on this community at risk. An ecosystem health approach recognizes the inextricable links between humans and their biophysical, social, and economic environments, and that these links are reflective in the population's state of health. The results shown here are the result of stakeholder consultation and represent real community concerns. Our approach uses community-based participatory research methods in which research questions and design were developed both by the investigators and by the community. It also spans multiple disciplines with researchers from anthropology, biology, and medicine working together in order to inform the ecosystem health concerns. It's also founded on the principles of sustainability with the idea of knowing what is practical and appropriate in the community is what will be sustainable. So as I mentioned, both real and perceived exposure to pesticides is a major concern in Naivasha. Our hypothesis is that the floriculture industry is a major source of stress and of pesticide exposure. In order to investigate this hypothesis, we utilize two biomarkers. Hair cortisol is increasingly being used as a biomarker of chronic stress. Circulating lipophilic compounds such as cortisol can be incorporated into the hair shaft. Unlike traditional matrices to measure cortisol such as blood, saliva, and urine, which only represent a moment in time, hair cortisol gives a picture of chronic stress. It is also an attractive medium as it's easy to collect, transport, and store, and it's measured with an enzyme-linked immunosorbent assay. We collected hair from 107 participants from Naivasha and from Mogotio, which is a city similar to Naivasha but removed from the floriculture industry. Inhibition of red blood cell acetylcholinesterase was used as a biomarker of exposure to organophosphate and carbamate pesticides. We collected blood from the same participants that we collected hair sampled and measured acetylcholinesterase in whole blood using a WHO-approved spectrophotometer field kit that measures asymptomatic pesticide poisoning. We also created and implemented a survey in order to determine socio-ecological indicators, including socioeconomics, employment, gender issues, water use and collection, living conditions, and migration. This survey was implemented to over 1,000 participants as a representative sample of Naivasha and Mogotio. We took a random subsample from these 1,000 in order to collect the hair and blood samples previously described. So in order to analyze this data, we used regression tree analysis. Regression tree analysis is the test in order to predict the value of an outcome based on several independent variables. It utilizes binary recursive partitioning, whereby the observations are repeatedly bifurcated into nodes based on the considered indicators for the predicted outcome. We utilize the Gini index, which maximizes node separation, ensuring homogeneity between the nodes. So this regression tree analysis shows the levels of cortisol that we measured in hair as the dependent variable. The levels seen here are in nanograms per gram, with the higher levels indicating more chronic stress. So the socio-ecological indicator that best predicted hair cortisol or chronic stress was sex, with females having higher levels than males. In particular, it was seen that females who are aged 50 years or older, a majority of these females do not work on the flower farm, had the highest levels. Males who do work on the flower farm had the lowest levels of stress. We could also see that age plays a major determinant in many of the splits seen in the data. With the second highest levels of hair cortisol was females aged 25 to 29, with more than 2 people living in their house, had the next highest levels of stress. And a majority of these women also do not work on the flower farm. For females aged 35 to 49, those that did work on the flower farm had higher levels than those that did not. The next regression tree utilizes the percent of depression of acetylcholinesterase as the dependent variable. So again, the acetylcholinesterase depression was used as a biomarker of exposure to organophosphate and carbamate pesticides. So 100% would indicate no depression or no exposure to pesticides, and a lower the number would indicate there would be more exposure. So the socio-ecological indicator that best predicted pesticide exposure was again by sex, with females having more exposure than males. In particular, females who do not believe they're being exposed to pesticides, who live in a non-permanent rented house, had the lowest levels of depression and hence the most exposure. Males who completed secondary school had the least amount of exposure. So for males, we could also see that education was a predictor of pesticide exposure, with those who have completed lower education having more exposure. We could also see from this regression tree that females who believe they are being exposed to pesticides and work at the flower farm have lower levels than those that don't work on the flower farm. So next we looked at the percent of acetylcholinesterase depression that I just described previously, and we broke it down by sex and by response to one of the questions we asked on the survey, which was, do you think you're being exposed to pesticides? So people who responded yes, they believe they were being exposed to pesticides, had significantly higher levels of acetylcholinesterase, indicating that they have less exposure to pesticides. So next we did some simple statistical analysis in order to determine some sources of stress in the communities. So there was no significant difference in hair cortisol content between the different communities in Naivasha and in Magotio. Volunteers from Naivasha and Magotio had significantly higher hair cortisol content than our reference group of Europeans. And lastly, we found significantly higher hair cortisol content in females, in participants who were divorced compared to single, married, or widowed, those who made below what is the minimum wage in Kenya, and those who reported feeling unsafe collecting water or using sanitation facilities. Looking further into the pesticide exposure, we found no differences in the level of acetylcholinesterase between the different communities in Naivasha and Magotio. However, when we looked at the different positions within the floriculture industry, we found those that are in the position of graders or harvesters. So these are typically women who sort the flowers and ensure that they're suitable for export, had significantly more acetylcholinesterase depression than sprayers who sprayed the pesticides or general flower farm workers, indicating that this group might have more exposure. So in conclusion, contrary to popular belief and to our hypothesis, it appears that stress and pesticide exposure appears to be more pervasive in the communities in Naivasha and not restricted only to the floriculture industry. We found from our analysis that gender is a major determinant of both pesticide exposure and of stress. This is likely due to the socioeconomic and cultural factors that exist that prevail in sub-Saharan Africa. This is particularly important to investigate as women are critical to the success of the floriculture industry, as well as to the running of the household and raising of the children, where women typically perform the largest share of labor. We also saw that both education as well as an awareness or fear of pesticide exposure actually acts as a deterrent or protective factor to actual exposure. So overall, we can see that socioeconomic issues appear to be of more importance for stress at Naivasha than pesticide exposure. All of these factors are important to investigate further, as an ecosystem cannot be sustainable until its most vulnerable members are protected and healthy. So I'd like to thank my supervisor, Dr. Jack Ben, and my co-supervisor, Dr. Charlie Trick, as well as my advisory committee. I'd also like to thank the members of the Lake Naivasha Sustainability Project, particularly Dr. Irina Creed and Dr. Pamela Zimbiri at Egerton University. I'd also like to thank the clinical sampling team, as well as our community representatives from Naivasha and Mogocho, and my funding sources, CIGI and the IDRC. Thank you.